As we begin moving through the microbiology course, we're going to start with just a little overview of the subject of microbiology and give you a little bit of information on what we're going to study throughout the semester. So as we start in chapter 1, the first term we need to think about is the term ubiquitous. The term ubiquitous tells us that microorganisms, also called microbes, are found virtually everywhere. As you can see in these pictures, this is just a few examples of the different environments in which you can find microbes. In the ocean, frozen in the Arctic, in the dirt, in the air, on your skin, microorganisms are found everywhere. In order to be classified as a microbe, the organism must be too small to be seen without a microscope. So as we look at these pictures, we can't actually see the microbe, with the exception of this picture here. This is a microscope image. But knowing that those microbes are there is really what is interesting about this subject. A lot of us may think about microbes as something bad. We think about bacteria or viruses that cause disease, but in reality, most microbes are completely harmless and can actually do good things for us. An example of some of the good things that microbes can do for us, other than creating oxygen, helping make fuels for us, microbes are used to ferment food and beverages. We would not be able to make things like beer, wine, bread, yogurt, cheese, for example, if we did not understand microorganisms and the process of fermentation. So as we go throughout the semester, we'll actually learn about fermentation and how that works. Let's take a brief look at the history of microbiology. There's a little argument as to where the subject of microbiology began, but most people can credit the first true discussions of microbiology all the way back to Hippocrates. Hippocrates did a lot for medicine, but what he did for the subject of microbiology was he was the first person to completely dismiss the idea that diseases were caused by supernatural forces. Now, Hippocrates had no idea what was causing the disease, but he knew it was not just an act of nature. Now, following this, Varro then said, you know what, I think there's something we can't see that's causing the disease. But he still was not the person that discovered microbes. Microbes weren't discovered until over 1,500 years later, when Leeuwenhoek, the true father of microbiology, developed what we consider the first microscope. Now, it wasn't actually a microscope like we know microscopes today. It looked more like this picture shown here. It was actually just more of a magnification set of lenses. And what Leeuwenhoek did is he looked at a drop of rainwater and he noticed very small things moving around in the rainwater. So Leeuwenhoek discovered small organisms. Still not exactly sure that those organisms were what caused disease, but he discovered the first microbes. Around the same time, Robert Hooke invented a microscope, and he is who we consider the father of cell theory. He discovered the first cell. But Robert Hooke was not looking at microbes. He was looking at cells within this cork. A few other important people in the history of microbiology would be Louis Pasteur. He was the person that discovered fermentation. And what he was trying to do with fermentation and later on pasteurization was figure out how could we kill the things that were making food and drinks spoil. So what he did, he took a flask with a curved neck, he boiled it. What we now know was occurring was boiling was killing most of those microbes. And then with that curved neck, once we boiled and killed all the microbes inside of the flask, anything coming from the air would get trapped here in the neck of the flask, and you would get no growth of microbes. So he considered this pasteurization. He was heating enough to kill to preserve what was inside of his flask. The next experiment he did, he boiled the same way, but broke the top off the flask. Now, with the exposure to air, microbes would enter into this liquid inside of the flask, and we would get microbial growth. So he was proving that there was microbes in the air that was leading to the spoilage of food and different types of beverages. Now, later on in his research, Pasteur also developed some vaccines, and we'll talk more about that as we go through the semester. The last notable person to talk to you about at this point in the course is Robert Koch. Robert Koch developed his postulates, the four postulates. And what this breaks down to is this man demonstrated that one particular microbe could be the cause of a disease. And so he actually took an animal that had died from a particular disease, and he was actually studying anthrax. He was able to purify from the blood of this diseased animal a microbe that we now know, Bacillus anthracis, the bacteria that causes anthrax. He could then take that purified bacteria, inject it into a healthy organism, this mouse. Then when the mouse dies, he could purify the same bacteria out again, proving that it was this one bacteria that was causing the disease. Now, as we continue through the course, we will talk more about Pasteur and Koch and some of your other important people in the history of microbiology. But in Chapter 1, we really just wanted to get an idea of some of those important individuals. As we go through our microbiology course, it's important to keep up with how we classify microorganisms. There are three main domains that we use to classify organisms in scientific studies. The three different domains are the bacteria, which is where most of our microbes we'll be studying are found. The archaea. Archaea are really odd microbes. And then the third domain are the eukarya. You have some microbes in this domain, but you also have plants and animals and us within this eukarya. So we're going to talk about this a little more as we go. When we're looking at how to properly name microbes, we in this course will use binomial nomenclature. To do this, we will need to know the genus and species of each organism, remembering that the genus is always capitalized while the species is not. And then you either need to underline or italicize the scientific name. So if we think of a common bacteria that we're all familiar with, Staphylococcus aureus. So you can see here in this first bullet, this is the proper way to write the scientific name. Underlined or italicized, genus only is capitalized. If you're going to abbreviate a scientific name, the second bullet here shows you the proper way to do so. The genus name is abbreviated with just one letter, the first letter, and then the species name is spelled out. This right here, third bullet, is absolutely not correct. As we look at all of these different microbes throughout the semester, in your brain you need to kind of keep an idea of the relative size of what we're looking at. So these pictures here on the image show you about the size of an animal and plant cell, just kind of a generic one, compared to a red blood cell, which is smaller. Mitochondria, even smaller, would be within the other cells. And then a lot of your bacteria are going to be about the size of a mitochondria that you would find inside of an animal or plant cell. And then as we go down, your viruses are going to be even smaller than your bacteria. So going back to those classification groups, our three domains, this slide is intended to be just a quick reference for you as you go throughout the semester on the characteristics of these three different domains. The most important difference that we'll follow up with in the next chapter is the difference between the domain eukarya and the other two domains. Eukarya contains eukaryotes, which have true membrane-bound organelles. Bacteria and archaea are both unicellular prokaryotes, which means they are organisms that do not have membrane-bound organelles. The main difference between your archaea and your bacteria are the type of material that make up the cell wall. And again, we're going to cover this more in the next chapter. Now, viruses I have stuck out here over to the side. Viruses are unique. They're technically are microbes, but they are not technically living. So now let's spend the last part of this first week going through just an overview of the different types of microorganisms, starting with the algae. Algae can be unicellular or multicellular. Their cell walls are made of cellulose, making them very similar to plants. Also, your algae are majority photosynthetic, allowing your algae to be responsible for producing the majority of the Earth's oxygen. What makes an algae different from a plant mostly is the fact that they can be unicellular, more simple, and they are microbes. Algae are important in our everyday life. They help us produce oxygen. They're used in the laboratory setting in the form of agar. You have agar in your refrigerator right now. You purchased it in your lab kit. But somewhere you may not think, algae are found in ice cream, salad dressings, lipstick, toothpaste, all sorts of things you use every day. Our next group of microorganisms are the protozoa. These are really neat and probably some of my favorite microorganisms. They can be unicellular or multicellular. They are highly motile, meaning they can move, and they're classified by which type of organelle they use to move. They can have cilia, flagella, pseudopods. Some of your protozoa can be parasitic, cause some pretty nasty diseases, but then other protozoa are going to be found in your drinking water. And if you've ever ran water from the tap and took a sip, you've drank a protozoa, and you're all still alive, so you're okay. The third group of microorganisms we'll study this semester are the fungi. They can be unicellular or multicellular. The microscopic groups include yeast and molds. We're not going to be studying mushrooms. We're studying the microscopic. All cell walls of fungi are going to be made of chitin, making them completely different from plants and algae. Almost all types of fungus are going to be free living and completely harmless. But there are a few types of pathogenic groups that we'll go through as we go through this semester. The largest of the microbes are going to be the helminths. Helminths are parasitic worms. They are included in the microbe group because they are transmitted from one host to another during a microscopic stage. So this particular picture, this is a several foot long tapeworm that was taken from someone. And then our last group of microbes are the viruses. As I said, they are not composed of cells. They're completely acellular. They are obligate intracellular parasites. All of your viruses are parasites. So just to close, the goal of chapter 1 was really to give you an overview of the different types of microbes and what we're going to be studying this semester. We'll really get a good start with our bacteria. Remember, those are the ones that do not have membrane-bound organelles, and then we'll move in through those different eukaryotic microbe groups.